Recording. And now we're going to go to mini lab operations. So let me tell you that the mini lab has to be very simple to use, but it has to be incredibly capable to use. And let me tell you that this same mini lab is used at the upper elementary level, the middle school level, the high school level, the 2-year community college level, and the 4-year university level. And at the university level, students are doing peer-reviewed scholarly research in the same mini lab that the upper elementary school students are using. The best way to explain this is that the sophistication of the experiments increases with grade level. And what you're able to do, the mental model, is you can put up to 3 small test tubes of fluids and solids into space. Space is incredibly expensive. To send 1 American astronaut to the International Space Station by way of SpaceX Crew Dragon is a bargain basement price compared to what we were paying the Russians. It's only $400,000 per pound. $400,000 per pound. $55 million on average to send 1 American astronaut to space and back. So we have to accelerate your experiment to 18,000 miles an hour. We have to put it 250 miles above the surface of the Earth. That's way expensive. So what we're able to do is get a break as a nonprofit to put three small test tubes of fluids and solids in space that can be combined in a prescribed way. This is the NanoRacks Mini Lab. It is a tube about 7 inches long that can be divided into up to 3 chambers by the introduction of clamps. The new Mini Lab is twice the volume, and it has screw valves that open and close. So 3 small volumes of fluids and solids that can be put up in space. Now let me give you another piece of information here. The technology of flight operations to and from low Earth orbit require you to submit your mini lab to Houston for payload integration 2 to 3 weeks before launch. And the launch could be delayed, which means that you probably don't want your flight experiment operating in Earth gravity for many weeks, which means you probably want to turn your experiment into Houston in an inactive state to be activated by the astronauts. And when it deorbits, it'll take a couple of days to get back to your flight team in Earth gravity, and your flight team might not be ready for harvesting and analysis right away. So you probably want to deactivate your experiment before deorbit. And that's why 75% of the experiments are biological in nature. 25% are physical and chemical systems, 75% are biology. And so the idea here is, let me give you 2 examples, okay? At lower grade levels, for instance, you have an infinite variety of dried seeds that can be hydrated on orbit. And at upper grade levels and at undergraduate levels, you have hundreds and hundreds of microbiologicals that are available in what we call a lyophilized form, a freeze-dried form that can be hydrated on orbit. And biologicals can be killed and preserved by introducing a fixative like glutaraldehyde or formaldehyde or maybe just alcohol. So does everybody understand why 75% of the experiments are biological in nature? Make sense? Okay, so let me give you 2 concrete examples. Hillsborough County, Florida, a middle school team put forward the following proposal. They said that astronauts are going to now go back on long-duration space missions. They're going to go to the moon. They're going to Mars. They're going to establish a moon base. They're going to go to asteroids. They're going to have to grow their own food. But we know that biology, all biology, behaves differently in a microgravity environment. Gravity plays a role in biology. So they asked a really cool question. I wonder if the germination rates for food crops is the same in a weightless environment, because if it's not, maybe the astronauts can't grow their own food. So in the first chamber, they put dried lettuce seeds. In the second chamber, they put distilled water. And in the third chamber, they put alcohol as a fixative. So once it got to orbit, the astronauts opened the first clamp and shook it to hydrate the seeds. And after about 2 weeks after seed germination, the astronaut introduced the alcohol to kill and preserve the seedlings. The same thing was happening on the ground in the control experiment operated by the student flight team. And when the flight experiment came down and they put them side by side, they could easily calculate the percentage of seeds that germinated on Earth and the percentage of seeds that germinated in space, and they could compare the germination rates. Just visual inspection, they could see if the root ball shapes were different and whether or not the seedling itself, the sprout, looked different. And under high resolution microscopy, they could look at the cellular level to see if there were cell growth differences. So do you see how that experiment worked? Does that make sense? At the undergraduate level, a team said, we don't know if salmonella is more virulent in a microgravity environment, which could pose a health risk to the astronauts. So in the first chamber, they put freeze-dried salmonella. In the second chamber, they put a Luria broth growth medium. And in the third chamber, they put a fixative called glutaraldehyde. So once it got to orbit, the astronauts hydrated the salmonella and the colony began to grow. They wanted the fixative introduced only 2 days later because they didn't want the colony to die in its own waste. So the astronaut then introduced the glutaraldehyde to kill and preserve the salmonella. Same thing was happening on the ground. And when the flight experiment came down and they put them side by side, this team in the laboratory could measure colony size differences very accurately. And under high resolution microscopy, they could look at morphological differences at the cellular level. So does everybody understand those two kinds of experiment? Okay. So anybody have any questions about
Yes.
mini lab operation?
Do you have a non-biological example?
Yeah, there are experiments in crystal growth and protein crystal growth. There are experiments in oxidation. And so I'll tell you in a little in the next chapter how students can read a summary of over 400 experiments conducted in this mini lab via SSEP. So does anybody have any other questions before we move on to the next chapter? We're good? Okay, what we'll do is, so Pam, you said that you'd like to end it in 2 hours, but there's no way to end in 2 hours. I don't know what to tell you. We can keep going until it ends, but it's not going to be 2 hours. I also want to give everybody, a few minutes break. So how do you want to handle this? We just keep recording?
Yeah, I think we can keep going.
Okay, so let me go to the next chapter. So I'm going to stop